For my project, I studied the effects of behavioral enrichment items on the stress of the ring-tailed lemurs and the black and white lemurs at the Trevor Zoo through behavioral and fecal analysis. Enrichment is a mental stimulation that allows the animals to prevent themselves from performing stereotypic behaviors. Stereotypic behaviors include pacing, vocalizing, excessive scent marking, or excessive grooming. The black and white lemurs were the only ones that actually were exhibiting these behaviors, but I included the ringtails to compare their stress levels to the black and white lemurs. And I decided to design my own enrichment item for this, so that way hopefully their cortisol levels would change more drastically. And I decided to design a wooden box with 3 different doors that had 3 different latches on it, so that way it'd be more of a puzzle for them and they'd have to really think about it, and hopefully it would change their stress levels, either increasing or decreasing. I also found that there's different types of stress, so even if their stress levels were to increase during this process, which they actually ended up doing, it's because of hopefully good stress rather than the bad stress. And for the process is I gave them small graded pieces of non-toxic wax crayons, so that way I'd be able to tell their feces apart from one another, so they each had their own color. And for the behavioral observations, I took ethograms, which are charts that have behaviors across the top and time increments along the side. And I did 30-second time increments, but depending on how active the animal is, you decrease the time or increase the time that you mark down. So I took these from outside the exhibit, and every 30 seconds I would scan the exhibit and see what behaviors they were doing, and then I would mark that behavior down. I found that the black and white lemurs exhibited pacing, vocalizing, and excessive scent marking. It was mainly Bombo, the male, that conducted these behaviors, and the female Maki and baby Billy did not seem to exhibit any stress through the behaviors. The ring-tailed lemurs didn't show many stress behaviors either. They just kind of were stationary for the most part and were grooming each other. For the feces, I collected samples every morning and every afternoon. So in the morning I would do it at 8:30, and in the afternoon I would collect at 4. And then I kept these in the freezer until it was time to do the assay. With help of Dr. T and Ms. Bennett, I conducted the assay, which we took the feces samples and weighed 0.5 of the feces and put them in test tubes, and then added 80% methanol to each of them. From there, we had to put them on a vortexer for 2 minutes to extract the hormones out of the feces and into the methanol. And then from there, we put it in a centrifuge, which is this machine that mixes it around even more. And we did that for 15 minutes, and that extracted all of the hormones, or hopefully all of the hormones, out of the feces and into the methanol. We then left those out for about 3 days, so that way the methanol could evaporate and the mixture would be more diluted for when we were running the assay. For the assay, we followed Enzo's cortisol kit instructions, and we basically had this small plate that had individual test tubes on it, and we put each of the 74 samples on it, and then we had a couple left over for the standard, so that way we could guess where the curve would be. On this graph, this is the results from the assay, and the black and white lemurs are on this side and the ring-tailed lemurs are on the right side. And the lines that are at the top, those ones were high stress levels, so we guessed what number that would be for the top ones, so that way it could show up on a graph. Their stress levels could be much higher than that, but the ones that are lower down are the ones that actually hit our curve. So Dr. T and I were thinking of retesting these over the summer and maybe just adding 0.25 grams of the feces, so that way hopefully the stress levels that were higher would actually hit the curve this time, and we could see how stressed those animals truly are. For Bombo, his is here. His were high without the enrichment, and then when the enrichment was offered, his went down. So his matched up with his ethogram, whereas the other ones did not, because Maki actually, hers is this one, she was the most stressed. but her ethogram and behavioral observations didn't show that she was actually stressed. But during this process, she was pregnant. So now we're thinking that because she was pregnant, her stress levels were like skyrocketing, because the red dot shows the day she gave birth, and the next day her stress levels went down a lot. So another reason we would retest these is we would take more fecal samples from later on, and we would see if females are just more stressed than males in lemurs, because the same thing happened with London and Lorelei, the two females with ring-tailed, where they were stressed all throughout, even without the enrichment and with the enrichment. So we were thinking of retesting them, because even though London, she wasn't the one that was stressed, but her data shows that she was even more stressed than Lorelei, who was the one that was pregnant. So we have the average of all of the stress levels, and these are the black and white, and these are the ringtails. So the behavioral observations do match up with the assay, since it shows that the black and white lemurs overall are more stressed than the ringtailed, and that's what the behavior observation showed. And this is just another graph of just the average of each of them. So we have Maki, Billy, and then Bombo, and London, Lorelei, Foster, and Jono. So Jono's the baby, so he had no stress, which matched up with his ethogram as well. And even with the enrichment, he didn't seem stressed at all. He just kind of was licking the boxes, actually, when I wanted him to open them. And then I think that's it. I have the pictures of what the bananas looked like when I fed them with the crayon on them. This is Billy interacting with the enrichment, and then Jono and Lorelei with their enrichment. And that's what the actual enrichment ended up looking like. But yeah, thank you.